Okay, my name is Jae HyungMah, a professor of Korean University. It's my pleasure to present my research outcomes at the conference on food processing and technology in London. Today, my speech is titled as Occurrence, Impact, and Biocontrol of Biogenic amines in fermented soybean foods. First of all, I'd like to introduce what biogenic amines are. They are actually very harmful or toxic nitrous compounds produced by microbial decarboxylation of amino acids in foods. So it includes several different biogenic amines, likely monoamine, diamine, and polyamines. Fortunately, our human body has detoxification system. So it includes monoamine oxidase, diamine oxidase, and polyamine oxidase, so that most of oral intake of biogenic amines doesn't make any problem on human health. However, if somebody eat high dose of amines, or if somebody is treated with specific inhibitors, likely antidepressants, then these biogenic amines can make a problem. In detail, the tyramine in food can be oxidized by the monoamine oxidase in intestinal tract. However, if somebody is treated with the antidepressant, a tyramine in food can directly raise blood pressure, so that it results in hypertensive crisis. Another case is for histamine. The human can make an allergic reaction if we are exposed to some kind of allergens. So you can see the allergic reaction over there, but the biogenic amines, especially histamine, is not related directly to the allergic reaction. However, if somebody eat too high level of histamine, it can lead to the very allergy-like reaction, so that it can affect the lung, skin, and intestine, or the other human organs. Furthermore, these biogenic amines can induce some toxical effects. Some of them is very similar with foodborne disease. However, there are several very important diseases are involved in this reaction. So that so many countries like the United States and European Union have their own regulations or the upper limit. Likely in Korea, the Korean government settled down the regulation very lately. So you can see there, I was on the air to announce the regulation in Korea. Furthermore, several authors suggested the maximum amount of biogenic amines in food. So in this case, histamine, tyramine are major targets to provide the tolerance levels, and which includes several different foods, especially the fish. And you can see there, several fermented foods are involved in this table. Then what about the fermented soybeans? The fermented soybeans have known that very important foods for human health. Actually, we are thinking that they are kind of functional foods. So we can easily overlook the risk of biogenic amines in fermented soybeans. That's why Asian ancestors have eaten these fermented soybeans for a long time. That's why we can easily overlook the risk of fermented soybeans. Like the other protein-rich foods, the soybean contains very high level of proteins, and it can provide amino acid precursors of biogenic amines. So if the soybean is fermented by microorganisms, the microorganisms can produce biogenic amines during the fermentation process, so that the biogenic amines can easily make the toxicological effect during the fermentation. So I focused on the fermented foods, including doenjang, 청국장, gochujang. They are all Korean traditional fermented foods. And natto, miso, they are Japanese fermented foods. And dubanjiang is a Chinese fermented soybean food, and also tempeh, that is an Indonesian fermented food. The reason I focused on these foods is that they are all fermented or contaminated by Bacillus subtilis. If the Bacillus subtilis has one of the decarboxylase activity, then it can produce biogenic amines. So I'm going to explain my research outcomes with 3 different content. The first thing is the levels of biogenic amines in the fermented foods. The second is how we can control or reduce the biogenic amine content in the foods. The third thing is the molecular studies. So if I can have a little bit more time, then I can explain this part either. First of all, the levels of biogenic amines in the fermented soybean foods. The tofu is the typical non-fermented soybean foods. So as you can see here, the most of biogenic amine levels are very low. However, only spermidine is very high level in the tofu. The reason is that the spermidine is very important and required for the growth of plant. It's like a hormone for the growth. In contrast, natto, the fermented food, contains very high level of beta-phenylethylamine and tyramine, and also spermidine and spearmine, the polyamines. And miso contains relatively higher level of histamine and tyramine. And doenjang, gochujang, they are Korean food. It contains, a little bit higher level of histamine and tyramine either. And dubanjiang, the Chinese food, contains very high level of beta-phenylethylamine. Actually, it was 6 times higher than tox dose of this amine. And tempeh contains higher level of histamine. So as you can see, it's kind of summary. The dubanjiang, tempeh, and natto contains very high level of beta-phenylethylamine, histamine, and tyramine respectively. The red line means the guideline suggested by Brink and the other authors. So I summarized the frequency of the fermented food containing the higher level of biogenic amines than the Brink's suggestion. So as you can see, the right values mean the number of samples, and the left value means the number of the samples containing higher level of biogenic amines, each amine, than the guideline level. So most of the fermented soybean foods contains very high level of biogenic amines, especially histamine, tyramine, and beta-phenylethylamine. So our study has been supported by the report of the other research teams. So in Asian countries, so many research teams are trying to analyze biogenic amines in different soybean foods. So I tried to make it clear why these foods contains that much high level of biogenic amines. Of course, physicochemical factors and microbial numbers can affect the number, the amount of biogenic amines in the food. But the most important reason was the bacterial activity. So even though the foods have the completely same number of microorganisms, if they have different activity, then the activity can affect the content or amount of the biogenic amines in the foods. So, of course, we can have several different strategies to control or reduce the biogenic amines, like the physical method or chemical method, or even the biocontrol method. But in the case of physical and chemical method can affect the taste or odors of the foods because they are all fermented foods. So only way to control or reduce the biogenic amines in these foods is you can just develop new starter culture, or we can modify the fermentation conditions. So several authors have reported that most of microorganisms related to these fermented soybean foods can produce the biogenic amines, which includes even lactic acid bacteria. So several authors have tried to reduce the biogenic amines by modifying environmental factors like pH or salt content of the food, or modifying the temperature during the storage or process, the fermentation of the food. All these have been reported in the late 20th century. And then several authors tried to develop or find amine negative starter cultures, which means that they cannot produce biogenic amines. But actually, the result is saying that only part of biogenic amines can be reduced by using these amine negative starter cultures. I don't know why it happens, but I think that they reported, even though they reported amine negative starter cultures, which means that a part of biogenic amines are negative, but the other biogenic amines can be produced by the same starter cultures. That's why they reported only part of the biogenic amines can be reduced by using these starter cultures. And most lately, some authors have tried to degrade the biogenic amines by using microorganisms. So I think it's the best way to reduce the biogenic amine content in the food. Now, then what do we have to do in the future? So my research team have tried to develop molecular techniques to reduce biogenic amines. The first thing I did was I wanted to see if there are genes related to the biogenic amine production in the Bacillus subtilis. The Bacillus subtilis is the most important microorganisms in the fermentation of soybeans. So on the NCBI website, I found two different genes. One is related to ornithine decarboxylase, which is related to putrescine production. Another one was lysine decarboxylase gene, which was related to putrescine production. By using this information on the genes, I amplified the genes in the Bacillus subtilis like this. The problem was that the most toxic biogenic amines are histamine and tyramine, but somehow I couldn't find any information on the NCBI website. So I'd like to give you an assignment here. So if you can find the gene for the histidine decarboxylase or tyrosine decarboxylase, that would be very exciting finding. So anyway, I successfully amplified the genes, and I applied the method to detect amine positive or negative microorganisms. So when I tested type cultures, so interestingly, some of the type cultures can produce the biogenic amines because I detected the genes, and part of the type cultures cannot produce the biogenic amines. So I thought that if some of the type cultures can produce the biogenic amines and the others cannot do this, we can easily select starter culture candidates by using this molecular technique. So I applied this result technique to the isolated strains from so many different types of fermented soybean foods. Then it was very lucky. So I found only one strain which don't have the gene of both of ornithine and lysine decarboxylases. I don't know why. I think that the strain might be mutated spontaneously, but I don't know. Now, I also try to use RT-PCR technique in order to see the gene expression level. Then the same strain, I found that the same strain has very low level of the gene expression compared to the other strains. After combining this result with HPLC analysis, I found very interesting situation. Even though HPLC analysis result can give us, it doesn't produce biogenic amines. The PCR result can give us, it can produce very high level of biogenic amines. So it doesn't match with each other. So I'd like to suggest that if we want to select amine negative starter culture candidate, we have to use both of HPLC and PCR techniques. Now, it's very famous article in the field of my research. Dr. Hojafer, the most famous microbiologist, one of the most famous microbiologists, announced that we have to develop genetically modified starter culture in the future study. So I wanted to follow his suggestion by knocking out the genes and developing mutant strains. So by using double crossover homologous recombination technique, I successfully construct mutant strains, which means that they don't have the genes of decarboxylation enzymes. So this slide shows that the mutant strains doesn't have the gene. Now, each the result of the cultivation of the strains. As you can see here, when I knocked out the lysine decarboxylase gene, the corresponding result says it doesn't produce cativaline. Likely, when I knocked out the ornithine decarboxylase gene, the mutant strain doesn't produce putrescine anymore. Furthermore, very interestingly, the other biogenic amines, the levels of the other biogenic amines also reduced by using the knocked out mutant strains. So I'd like to summarize my research outcomes. First of all, actually, the amount of biogenic amines in most samples are within the safe levels, but several kinds of biogenic amines in several types of fermented soybean contains very higher level of biogenic amines, and furthermore, some of them was greater than the toxic doses. The second thing is why they can be different levels depending on the types of the fermented foods. They must be because of the ratio of ingredient or microbial activity or the conditions or period of entire food supply chains. So in order to control and reduce the content of biogenic amines, we need to use starter cultures. For example, amine negative starter cultures or amine degrading starter cultures. The third part is, if possible, we can use molecular techniques to detect or to analyze the expression levels of decarboxylase genes. It would be a very successful way to reduce the biogenic amines. And for the future production of fermented soy foods, I'd like to suggest that we need to prepare and develop knockout mutants without the related genes. I don't know if European countries allow this kind of starter cultures, but of course Korea doesn't allow this technique. But someday later, likely 10 years or 20 years later, we may need to try to use these starter cultures if we can design the genes very well. So these are key publications of my research team. during the past five years. And my stunts, the reason I'm here is one of my stunts is from Finland, so I like Europe. Thank you very much for your listening to my presentation.
Thanks very much for your informative presentation.